Okay, so I'd like to welcome our next speaker, Dr. Bruce Ritchie. Dr. Ritchie is a professor of medicine and hematology at the University of Alberta in Edmonton, where he is active in both clinical and basic research. His clinical interests include primary immunodeficiency, hereditary angioedema, hemoglobinopathies, and blood-borne pathogens. Dr. Ritchie developed specialty clinics in Edmonton to provide comprehensive care, including home treatment and blood products to these groups. He is also intimately involved in surveillance of the blood system in Canada, among other important endeavors. And Bruce, you've been such a champion of Immunity Canada for so long. And I just want to say thank you so much. And we're really excited to your presentation. So welcome.
Just this button. Okay, yeah, sure. Thanks. Cool. Oh, sorry. Thanks, David, for that kind introduction. Can you guys hear me? And I wanted to say thank you to Andrea. That was a lovely presentation. And it took me way back 30 years. I actually have been in Edmonton 33 now. In fact, 33 and a third. Third of a century has been spent in Edmonton. But way back 33 years ago, one of my colleagues in Calgary, a guy named Tom Bowen, did Canada's first human gene therapy experiment on a kid with adenine deaminase deficiency, ADA deficiency. The family had had one child already with ADA deficiency, which was before the days of newborn screening for this problem. And so the kid wasn't discovered at birth and unfortunately died. So when they had another pregnancy, Tom arranged for them to have cord blood taken prior to birth. And he sent the cells down to California where they were doing a trial on gene therapy. Unfortunately, 33 years ago, the technology wasn't very good and it didn't work. And unfortunately, that kid died as well. But you know, I think Calgary has had a long history of, and Alberta in general, you know, we have a history of trying to pioneer things. You know, we're often thought of as rednecks out here, but I think we're not. I think, you know, the West, the prairies in particular, is the home of social responsibility. It's where all the major social movements in the country started, you know, whether it's the CCF, which became the NDP, whether it's healthcare, which started in Saskatchewan. Alberta actually had universal healthcare as a provincial program before Medicare came along. So I think we are different out here. And those guys that think we're rednecks, we'll let them stay in the East. May they freeze in the dark, as we used to say. But anyway, I've been involved in immunodeficiency since about 2000. And at that point, I had been doing molecular biology and molecular medicine, trying to get it off the ground, trying to do gene therapy in hemophilia for a while. And we thought, when I came back to Edmonton in 91, that gene therapy was right around the corner. You know, it seemed so tantalizingly close. But sure wasn't close at all. And a decade on, you know, we were no closer than we'd been when I started. And so I pivoted from basic research to more clinical research and started doing more clinical work. And one of the things I did was to say, hey, these comprehensive clinics that we've been running for hemophilia are really good. And why not do them for the other disorders? At the same time, some of the patients with hereditary angioedema or HAE and also immunodeficiency had started arriving on my doorstep and saying, why can't we be on home treatment like those other hemophilia patients? And my answer was, I don't know, why not? And luckily, our hemophilia nurse also believed that. And so she came down and got things going. So we just with those couple of early clinic visits, moved on and developed these fabulous rare blood disorder clinics. We've really pioneered this in Canada. Calgary is a little bit behind us. They have similar clinics, but they're They're catching up to us quickly. Really, the rest of the country doesn't do this. They do it for hemophilia. Our hemophilia clinic started in 1980 or 77, 78. But the rest of the countries, so our hemophilia clinic started in 77, 78, and then our rare blood disorder clinics started in 2000. The rest of the country has hemophilia clinics, but no rare blood disorder clinics per se. And the advantage is we're able to support home care and a lot of things that many of the other centers aren't. We now track 1,000 patients with immunodeficiency out of Edmonton, and we have 500 on home subcutaneous immunoglobulin. And we think that's a big advance. So anyway, that's sort of me, and I'll just give you an update on what I see happening in the world. You know, we're in a time of unbelievable change. You know, I don't know if that's just because I'm old, because I remember my parents saying that, you know, 30 or 40 years ago, they couldn't believe how quickly things were changing. But I think it's moving at light speed now. Huge, huge, huge breakthroughs in medicine, huge breakthroughs in technology. And if you just forget the politics and some of the social media stuff, you can put your head down and ignore that. You know, the world is really going in a good direction. So I wanted to just talk a little bit about the changes in knowledge that have happened, and really point to a couple of the latest ones that we've been very interested in. One is about protein structure. So most of these disorders are disorders of proteins, whether it's immunoglobulins or other things. And in 1994, the Nobel Prize this year in chemistry, so just a few months ago, was won by Demis Halabis, I think. Anyway, he's a Greek Cypriot who was raised in London and is really a superstar. He's made advances in computing science, but also in biology. He had a seminal paper about 10 years ago that changed the way we think about cognition, for instance, as a neuroscientist. So very interesting guy. And he developed this company called DeepMind in London, which is really the first artificial intelligence company. It's done great work. And one of the projects he did was called AlphaFold. And AlphaFold made a big breakthrough just 4 years ago in 2000, when it took part in a competition to predict from the sequence of a string of amino acids what the three-dimensional structure would look like. We had technology back then to figure that out using X-ray crystallography and NMR, but those projects would take, those technologies would take at least a couple of years to solve a structure. And so we were probably closing in on hundreds to maybe 1,000 structures when he came along, and he said, well, that's not fast enough. I think artificial intelligence can do this. And so in 2019, I think, they entered a competition to predict structures of a protein whose crystal structure was about to be solved. And they came in first, but they were about 90% correct. Came back a year later in 2020, and they had nailed it. They had as good a structure as the crystal structure was solved on a subsequent protein. And so obviously the next thing people started saying was, well, if you can do that for one protein, why don't you do the rest? And so over the next year, they predicted crystal structures on 200,000 known proteins. And those are all the proteins that we know that exist, not just in humans, but every species that they could come across. So 200,000 structures. So we went from, you know, taking 2 to 3 years to solve a single structure to getting all 200,000 that were currently known in a year. So, you know, that breakthrough is huge. And they just gave that away. They just said, okay, everyone, here's the structures, do what you want with them. And so, you know, obviously that does win a Nobel Prize. It deserves a Nobel Prize. And we started using that program. And their next iteration, which looks not just at how the proteins fold, but how they interact together, what we call protein docking. And then just last week in the BBC, where I get most of my science news from, There was a story about the Human Cell Atlas, which was published for the first time this month in Nature, where they published 40 new articles on the Human Cell Atlas. And what they've done is, you may not know this, but we are made up of about 37 trillion cells. That's a lot of cells, okay? Some of us have more cells than others. Big people have more cells than littler people, but it's a lot of cells. On average, about 37 trillion cells. And each cell has about 3 billion nucleotides in the DNA that code for all the proteins that the cell makes. And of course, all the cells have the same DNA in them, but they all end up with different structures and different functions. And so how does that happen? And so the Human Cell Atlas was a project that was developed to try and answer that question. And it's being done by 36 different groups around the planet in 103 different countries. And basically what they're doing is looking at collections of cells, whether they're eye cells or nervous system cells or kidney cells or whatever, and trying to figure out what the proteins they make are, what the genes are that are turned on, and all that sort of thing. And they've been working on this for about a decade now. I've been watching this with interest. And then just this past week, they published their first iteration. And they have data on all the systems in the body. Oh, first of all, this is the guy from AlphaFold. And this is what he was doing. So proteins are a structure of consisting of a string of amino acids that somehow fold into a three-dimensional structure. And the key three-dimensional structures are called beta-pleated sheets or alpha helices. But predicting which of these fold into which and which are just loops between them was the problem that his group solved. And they could figure out a three-dimensional structure like this. They've gone on to make a version 4 of AlphaFold, which is looking at the docking of two proteins. It's not working very well yet, but we've been doing some work with it, and I'll show you that in a minute. And this is another paper that he published in 2007, deconstructing episodic memory with construction. So figuring out how memory works. And this was really a breakthrough paper. So just stay tuned. You know, there's, I think, 3 people who've won more than 1 Nobel Prize, and I think this guy might be one of those. And this is the Human Cell Atlas, excuse me. That was just published the 20th of November. What's the date today? 23rd, I think. So only 3 days ago. Huge breakthrough. And this is what it looks like. Oops. All the tissues they've looked at, and this is the number of cells they've actually got information on, whether the genes are turned on or off, which genes are turned on or off, which proteins are made, what they look like under the microscope, all those sorts of things. And you can see the different number of cells that have been studied. Okay, thank you. In the website for this, if you want, you can go to the website and it's available for everyone. And for instance, if you wanted to look at the immune system, you can see what sort of work they're doing and where they've got to. It's a very preliminary atlas. It's sort of like, you know, back in the day when first maps of the world came out, they had huge areas of the world that were not occupied by anything except the term "here be monsters." So this is the level at which we're at now, but it's a huge start. And this is just to show you where the data is coming from. So from all these different countries. So if you think the data is all going to be about rich white men, nope, it's about people from all over the planet, and it will be representative and will hopefully show us what the differences are between different populations. Now, I wanted to briefly touch on what's happening in Edmonton. You may not know about it because when you come to the clinic, things don't always seem to have changed much. You know, you come, you get your product, you go, you see the same old people wearing the same old clothes. You think nothing's much has changed, but I can tell you, over the last few years, huge amounts have changed. So one of the things was that we were trying to figure out what the genetic cause of these rare disorders we were dealing with was. And I'd been trying for about a decade to figure that out, and to get the genetics group in Edmonton to let us send our samples off to get genotyping done, find out what the mutations were causing disease. And I'd pretty much given up on that and a few other things. And then one of our new colleagues joined us, Adil Adatia. Have any of you guys come across him yet? Anyway, he's one of the treaters of the immunodeficiency clinic in Edmonton. He said, well, we should be doing genotyping. I said, yeah, good luck with that. So he applied and they let him do it first one off. And so what he was able to discover was this secret that they decided to open it up to everyone. So that was about 2 years ago when we started genotyping all the patients and trying to find out what the genetic cause was. And because I fancy myself a basic scientist, we've been taking that data and trying to figure out what it means biochemically, genetically, etc. And we've started doing protein modeling using those tools like AlphaFold, which works very well and has helped us a little bit. But we're trying to use the version 4, which shows us how the proteins that we're finding that are mutated are interacting with other proteins using protein docking software. And I can tell you it's fun. We're at the real bleeding edge on that and getting some things that absolutely look crazy and probably are. And I'll show you a little bit about that. We've also on another track, we've been working on blood product tracking, as David mentioned earlier. And for a long time, and those of you who are on immunoglobulin at home, you'll know we force you in Edmonton to use the CBDR app to track your product use. And that's because we completely believe that we need to be able to figure out who's got what lot number in case there's a recall, which still happens about once every 6 to 12 months. Usually they're for pretty minor things. But, you know, I was around in the days of HIV and Hep C when we were trying to do recalls with paper records and we couldn't figure out which lot numbers were where, who had what. And then when we did find out our patients had HIV and Hep C, and there was a compensation program going, we still had to go back through the paper records and figure out who got what lot number and would qualify for compensation. So clearly that was not a good way to do things. And with technological breakthroughs in databases and the internet and all that sort of thing, it's now possible. CBDR is the Canadian Bleeding Disorder Registry, and it was developed for hemophilia. And I helped develop that way back in 2000. I actually hired I had 2 summer students who drove their car from Victoria all the way to Prince Edward Island, stopping in 24 different clinics and installing software for a program we called CHARMS, which was before CBDR. And CBDR came along about 7 or 8 years ago, which was a big improvement because instead of us doing the data entry, we got the patients to do the data entry on their phones, which made a huge difference. And we were looking at the records instead of once a year, records provided by patients who were sitting in the waiting room filling out a year's worth of records on paper. Instead of that, we had suddenly real-time records and we could see what was going on and if there were problems and things. So it's a big breakthrough. We've been using this in Edmonton for all the products, whether it's immunoglobulins or C1 inhibitor for HAE or whatever. We're really the only clinic in the country doing that. Calgary started doing it with HAE, haven't started doing it with immunoglobulin yet. And I've tried to work with the hemophilia docs to make this a much more broad project, but they pretty much ignore me. And the more I push, the more they ignore me. But I believe in product tracking, and I've had this image in the back of my head that I'll show you based on a program from MIT, the Massachusetts Institute of Technology in Boston. And in that project, they had an idea way back in 2000 that we should be able to track the supply chain of any product that any company wanted to track using standardized identifiers like the barcode. And so I've had this in the back of my mind for the last 24 years. We've been trying to get the companies producing these factors to put the barcodes, standardized barcodes on, and they've done that for us. It took Health Canada about 15 years to get around to requiring it. But in the meantime, we are able to get the companies to commit to this along with Canadian Blood Services. And more recently, the FDA, the Food and Drug Administration in the US, and the European regulator called the EMA have brought in regulations that require not just the standardized barcode, which identifies lot number and expiry date and potency, things like that, but also a serial number on the smallest package size, whether it's a pill bottle or a vial or an individual syringe. And so that's been rolled out about 5 years ago in the US and in Europe. Europe went a little bit further and said, in addition to that, the companies have to put the data on every new product into a centralized database, and that's being developed in Europe. We believe the same thing. We believe that that would function much like the model from MIT from 20 years ago. And so when we've gone back to the companies and said, we want you to put a serial number on your boxes and your packages, they've all said, sure, it sounds like a good idea. We're eventually going to get around to doing it in Canada. But why? What are you going to do with it? And I would sort of do some hand waving and say, well, CBDR might be able to take this information. But they kept asking me that question. So we built a new version of CBDR, which we've called the Rare Blood Disorder Registry. And we're doing beta testing on that in Edmonton right now. And it's very much like CBDR on steroids in that you can read the barcodes with your smartphone with it. The data goes into a centralized database that's secure within Alberta Health Services. And we're now working with the companies and Canadian Blood Services and others to try and pull together this centralized model. And I'll show you that in just a second. I'm going to go back, but that blood tracking module also fits into some work that I hope Dr. Suresh is going to talk about, which is the CIEN registry, which is an immunodeficiency registry that's been developed with Dr. Nicola Wright in Calgary at the University of Calgary, and is getting very close to being ready to go. So this is some work we've done in Edmonton. One of my students, Lydia, you can't see her name down there, unfortunately, Lydia Navaratnam, took the information from our genotyping to look at the mutations in a protein called MTHFR1, which is a protein involved in folate metabolism. And she found that one of the mutations replaced a charged side chain amino acid with a much larger uncharged amino acid. And you can see just structurally, this is probably going to run into some of the chains from nearby. That's this mutation up here. A second mutation right here actually cut the molecule in half. This is actually made up of two domains that are held together by a loop here. You can't really see it, but this mutation means that this top domain is made and not the bottom part. And so it looks a little bit like this. This is the top part, and this shows the normal amino acid side chain here, and this is the new one bumping into the side chains nearby. And she did a little mini thesis on that project as part of a biology project. And this is the other mutation. The first one was up here. The second one truncates the protein by taking what we call a nonsense mutation, which shifts the frame. So as you know, proteins are made up of amino acids, and each amino acid is coded for by 3 nucleic acids in the DNA. And so if you either drop or add a nucleic acid, it shifts the frame that is being read by the gene transcription information machinery. And that's what happened here. And we added these extra, I think, 8 amino acids, and then it comes to a stop codon and stops here. So it just makes this upper part of that molecule. And Lydia is now doing further studies on the side as she works at a restaurant to make the money to continue to do this work. Really, it's quite amazing. This is work done by another student of mine, Ranver Minhas, and this is really the AlphaFold version 4 project that we're working on, showing the docking of this one protein in green with this other one in blue. And this is the native one, and this is the mutated one in which one amino acid is changed. And you can see there's quite a dramatic shift in how these are interacting with each other. We don't know if this is real or not. We're trying to do studies to try and figure that out. But I've been super lucky. I've got about a dozen undergraduate students who've been knocking on my door for some reason. You know, as I'm an old guy, turned 70 last week, and I'm trying to slow down and retire. All these students are knocking on the door saying, hey, can we come and work with you? We hear you're doing cool stuff. So Ranbir is one of them. He's done a great job on this. The mutation he's looking at, you know, normally when we think about mutations, we think about them knocking out genes like the ADA gene, as Andrea mentioned. But some of the mutations actually cause a gain of function and cause the protein to do additional things. Those are called gain of function mutations. And this is a very quick review of our rare blood disorder program. We've called it the MyCap blood product tracking. And MyCap is really the smartphone version of REDCap. REDCap stands for Research Electronic Data Capture. And it's a cool little program developed at Vanderbilt University to give industry level electronic data collection to individual investigators. And it started out to help a couple of investigators at Vanderbilt University, and it's now being used in thousands of installations around the planet by millions of users. And these are some of the students who are working on it here. Jack is an undergraduate student whose grandfather is a friend of mine, and he was out biking with me one day and said, hey, my nephew is looking for, or my grandson is looking for something to do for the summer. Can you keep him out of trouble? So Jack came and worked with us. Devin Reddy is an interesting guy. His father, Suresh, worked in my lab 30 years ago when he was trying to get into medical school. And Suresh has gone on to become a nephrologist and move into a house a block away from us. So we've been close to that family for a long time. And I've watched Devin grow up from being a little boy to the point where he calls me Uncle Bruce. And he did a presentation in front of Canadian Blood Services and Alberta Health Services and Alberta Health. And he kept referring to me as Uncle Bruce, and he couldn't figure out why people were laughing at him. Because he's of Indian extraction. We don't look much alike. Brian Kostlak is a computing science student who, I'm not sure how he came to work with us, but he's a superstar. Pranavi Thoda and Haley Rana are all, you know, they're just fabulous students, top-notch students doing great work. So this is REDCap, and this is what, if you look for REDCap on the internet, this is one of the pages that you will see. And up here it says, unfortunately you can't read that, but 7,621 installations. This is just from today. As of today, there's this many, 159. Oh, good. Oh, look at that. Who knew? But 159 countries, 2.2 million projects, 3.4 million users, and 42.8 thousand citations. In other words, papers written with this tool, because it's such a useful and inexpensive tool and globally available. And this is maybe how do we go to the next one now? Now we broke it. Oh, there we go. Okay. And these are the installations around the country, so you can see it's being used in lots of places. And one of the key things about this is this development of the standardized barcode. So back in the 1970s, a group in the US said we should be able to track merchandise easier with barcode. And they came up with something called the UPC. You might remember that, the Uniform Product Code. And the group that came up with this, basically, this barcode is like a foreign alphabet. If you were smart enough, you could actually read this, you could figure out the alphabet. But it's just a series of numbers and letters, information in a standardized format. And in it is the GTIN, which is Global Trade Item Number. So every product has a GTIN. So whether it's a chocolate bar, which is a small one, or a big one, they have different GTINs. And they're all the same. All commercial products on the planet have the same barcode, the same GTINs. And so if you buy a pack of Juicy Fruit gum in Tokyo and bring it to Edmonton and they scan it, they won't be able to read the Japanese lettering on the pack, but they'll be able to get all the same data off this barcode. And clearly, if that information is standardized, it's much easier to track it. In 2005, the FDA and EMA in Europe required that all pharmaceuticals, which includes plasma products like immunoglobulin, have a standardized barcode on it. Canada took about 15 years after that to get around to it. And so that's been a big breakthrough because it means you can scan these products and find out more information about them. Now, this is a picture that I found, and I'm pretty sure it was in a trade magazine back in 2000. And I thought this was such a good picture that I took a picture of it, and I've been using it ever since. I've tried to find that magazine again, but I can't. So I can't give credit for this, but it comes from MIT. And the idea is that you have a centralized object naming service, which is basically a server. And Acme Pizza Company up here says when it makes a new lot of pizzas, and then when it's is sold to a distributor, they can go to the object naming service and say, hey, we bought this from the company and we now have it. And then when it's sold to the store, they put the information in. They're exchanging this standardized information, which of course with the GS1 standard data is easy to do. And their idea from 24 years ago was that when you then took it home, you could actually scan it into your own inventory in your freezer. And I don't know about you guys, but we have a big chest freezer in the basement, and I have no idea what's in there. And so if we decide we're going to have a pizza one night, we just go and rummage through it and try and find a pizza, and we take the first one that we find. But you can imagine if you had a little database on your phone that said, yeah, you have 3 pizzas in the deep freeze, and you want to use this one because it's about to expire. That would be really useful, wouldn't it? So that was what their idea was. And then when you put it in the microwave, if the microwave could scan the barcode too, it would not let you guess what the directions were, but it would say, okay, cook this on high for 2.5 minutes, and then keep it cooking at low for the next 2 minutes after that. And you wouldn't have to know any of this stuff. And then when you threw it in through the box in the recycling bin, it would scan it as well and send a message to Acme Pizza Corporation to say, make another one. And if you think about this, Dell Computers has been trying to do this sort of model without this central server for a long time. They only make your computer when you order it. So if you go to the Dell website and say, I want this computer, they'll make it. But they don't have a warehouse because they don't want us to pay for a warehouse. So our idea was to try and use that, and this is what it looks like. This object naming service, we're trying to get GS1 to build it. In Europe, they're building it themselves. The European Union is building one. This idea of an object naming service has been around since the beginning of the internet. So whenever you guys get a new phone or a new laptop or a new computer and hook it to the internet, your computer says to you, what DNS server do you want to use? And most of you say, I don't know, right? So you hit the default one, and that's what we all do. And what you don't know is what the DNS server is. And it's a domain name server. And what the domain name server does, and it's been around since the beginning of the World Wide Web, is when you put in your web browser a name like apple.com, it goes out to this DNS server and says, what is apple.com? And it comes back with the URL, which is the string of characters that tells you where to take, where your computer has to go to find that web page. And you know, that happens within milliseconds. It usually happens before you're done typing, right? So this idea of having a DNS server has been around for the last 30 or 40 years. And it works quickly, and there's many copies of it on the internet. What we are proposing is that we do the same thing so that everyone can track the product. So when the manufacturer makes it, they put the information here. When they sell it to CBS, they do that electronically. Long before they actually distribute the product to CBS. But they say, well, we've sold you these million units of immunoglobulin, for instance, and these are the lot numbers and the serial numbers on them. And when CBS gets them, they check it to make sure they got what they thought they bought. And right now, we're working with CBS to do a pilot project on distribution of products through Bayshore instead of hospital blood banks. I'm not sure if you guys have had much experience with that. The pilot study is mostly complete, but it's going ahead. It's mostly with hemophilia products right now, and we're trying to expand it to subcutaneous immunoglobulin. But anyway, we're hoping to get the pharmacy service, Bayshore, to, when they distribute it to you, to actually send you a copy of what they're giving to you as well electronically. And when you use it, or when you get it, you don't have to add the inventory yourselves as you do with CBDR. And then when you use it, you would use it from a drop-down menu or from scanning it with your smartphone. And that would tell us that it's being used. And you might say, well, why do all this? Why worry about all this? One of the reasons is counterfeit medications. And you might say, well, what sort of problem is that? Well, it turns out to be a huge problem. So when COVID-19 was a big problem for us all, I read a story that 1 million doses of COVID-19 vaccine, which were counterfeit, were discovered in Singapore, ready to be shipped somewhere. And it turns out that with high value products, like, you know, a good example recently is Ozempic. You know, if you want to go on Ozempic and you can't afford it and your insurance won't provide it, you might buy it on the internet and you get a cheap one, like buying stuff from Temu in China, right? Problem is, a lot of that stuff is not real. And so you might get a vial with a clear liquid, which, if you're lucky, is sterile water or even plain water. Hopefully it's nothing more sinister than that. But you might get something that's fraudulent. What this allows you to do by using our RBDR here is you can go to the object name server and you can find out if this lot number in this product was actually made. I'm almost done, David. I'll be done in 1 minute. Whether this product was actually made, and if so, was it distributed to Canada and then on to Edmonton? Or was it made, but it was distributed and used in Brussels 3 weeks ago, in which case either they or you got a fraudulent one? So you could check this anonymously. And there's new tools being developed that will allow you to do this without sharing any information. And when you check it, the server will say, yes, somebody's checking it from Edmonton, but somebody also checked it from Brussels 3 weeks ago. So there's a problem here, and they can report it instantly to the authorities. Like before you even have finished scanning, a message will have gone out to say, hey, there's a problem here. We've got to track this down before anyone gets hurt by these fraudulent problems. And I think that's it. I think this is my favorite cartoon. As you know, in research, we keep telling everyone how good we are, when in fact, there is so much we don't know. There are way more unanswered questions than there are unquestioned answers, and there's a lot of unquestioned answers too. Okay, thanks very much. I'm happy to answer any questions.
Yeah, are there any questions in the room or any questions online? And if Bruce, there are, if you could just repeat the question.
Sure.
So people online can hear.
Yes.
Where are we at with any kind of protection in plasma products? Like I know we've always been about 18 months behind, but where are we at today in terms of like COVID-19 or virus protection? Do we have any information on that?
Yeah, the question is what sort of protection is provided by the immunoglobulin you're getting? How long does it take really from the collection of a unit of blood till the immunoglobulin, it's easy for you to say, the immunoglobulin from that donation appears in a lot number that you're able to use? And the answer is 18 months. So, COVID-19, people would say it started in 2020, but in fact, I remember in December of 2019, I started seeing the reports in the news from Wuhan saying there's this big problem. It's gone through China, and my spidey sense was tingling, and I said, this is going to be a global problem. And I started telling all my family who thought I was fear-mongering. I told them, don't press elevator buttons, use your elbow. Don't hold handrails. And the worst thing is escalator handrails. Don't ever touch those. And they laughed and laughed and laughed at me. And of course, 6 months later, we were in the middle of a huge global pandemic. But that was 4 years ago now, at least 4 years ago. And probably actually closer to 5. So the collections of plasma that were being collected were, of course, from people that may have had COVID-19 back as far as December of 2019. So, we've had some protection from that for at least the last couple of years. But it's not good enough, as you know, because you still can get COVID. So it's really, really, really important to get the COVID vaccines. Now, and that brings up another question of what about vaccines? We're often told as immunodeficiency patients not to get vaccines. Well, you should get vaccines, but not live vaccines. And the great majority of vaccines are not live vaccines, and certainly the COVID-19 one is a not live one. So you should get those. And you should get the flu vaccine, and you should get whatever vaccines you can. Now, I am the opposite of an anti-vaxxer. I'm a huge believer in vaccines, not just because they protect you against what the vaccine's all about, but I believe if you keep stimulating your immune system, it's not going to get old as quickly as it would otherwise. So one of the problems, and another biological thing I'm interested in, is called senescence, like why we age. And one of the reasons people often die later in life is because they get infections, sometimes with a bug called pneumococcus that causes pneumonia. And, you and I might get it and be a little sick, but I, as a 70-year-old, might get it and die because my immune system is a little bit old and not very responsive. And so I think if you keep stimulating your immune system with vaccines and other things, then it'll keep it young and healthy. So there's a lot of reasons why you should get every vaccine you can, whether it's the pneumococcal vaccine after you get into your 60s, or you guys should be able to get those vaccines anyway. All those vaccines, get them. Sorry. Yes.
I wonder what the study that you were part of, reviewing the efficacy of the vaccine on immune cases.
Sorry, say it again.
The study that you were doing with efficacy of vaccines on immune.
Oh, yes. Cases? Yeah, yes. The results of that, sorry, of course. We did a study in Edmonton. We were part of a national project to look at how effective the COVID vaccines were in people with immunodeficiency, and particularly with a type of immunodeficiency called common variable immunodeficiency. The thing about common variable immunodeficiency is it's very common and it's very variable. There's at least 100 genes that have been implicated in causing that disorder. So it's a hodgepodge of different disorders. And many of us, particularly me, we're not particularly good about classifying CVID. So we got a lot of data about a lot of different immunodeficiency states. And what we can say quite clearly is that the vaccines work. They work less well in people with immunodeficiency, but they still work in the majority of people. So you should still get it because it gives you some protection. Yes.
Thank you very much for the presentation. I have maybe a more simplistic sort of question. Right now with our SCIG logs, it's really just kind of manual data entry. You kind of take a picture of it and email it to the organization, and then assuming they manually data entry it. Did I hear you correctly that there's going to be a plan to directly enter that information?
Yeah. So you're obviously not from Edmonton.
No, I'm from Calgary.
Yeah. So the question was about using paper logs for tracking blood products and then taking a picture of it and sending it to the clinic. In Edmonton, we abandoned that, long time ago because that just wasn't working. Back in around 2000, we were collecting the data from our patients from the previous year when we saw them at the annual assessment clinic. And like I said, the patients were often found to be sitting in the waiting room, filling out a year's worth of logs in one sitting. Clearly, a good example of the garbage in, garbage out phenomenon. So we started a project called CHARMS, in which they had to give us their logs every 3 months minimum to get products. And that was a little bit better, but the paper logs then had to be entered into a database by our data entry people. That was a nightmare. And we were always weeks, if not months, behind. So the CBDR program that was developed for hemophilia and we used for everything else basically is a smartphone app that's been available for probably 9, maybe 10 years now. Basically, when you pick up your 3 months worth of product, you're supposed to enter that information into your own home inventory. And then when you use it, you go to a drop-down tab, which gives you a choice of which lot number and things you're using. And all that data is instantly shared with a central database that's kept in Hamilton and McMaster University. So we've been using that for a while. Obviously, Calgary hasn't been. We've been trying to encourage them to do it. And we're now going to replace that with a non-hemophilia product, which will do essentially the same thing, but you will be able to scan the products in with your own barcode scanner from your phone, just using your smartphone camera. And the cool thing about that is if they have a serial number on it and you think you can scan the same one 20 times, it won't work because they'll all have different serial numbers. So it's a way of making sure we have accurate data. And when you scan it in, we'll know that it's in your hands. And when you scan the fact that you've used one, we'll know that you've used it.
Right? And that will help in research too as well.
Well, and again, I'm hoping Dr. Suresh will talk about this CIEN registry, which is something we're hoping to feed our data into. So as part of this, this project I've discussed, the Rare Blood Disorder Registry, is a clinical tool. So we will tell you, well, I do tell people that they have to use it if they're going to go on the home treatment program. If they say, well, I don't want to use it, I'll say, well, that's fine. You can come into hospital and get your product. But if you want to be on home treatment, you've got to do this. That's my rules. And my colleagues aren't as hard-nosed about this as I am. So when one of the patients is giving us a hard time, the nurses call me, and I'm the guy that says, hey, I do it nicely. We're going to pull you off the program unless you do this. And I'm sorry, but it's important, right? So it is important, and it's a clinical. The registry, however, is more of a research tool, and we will consent patients to say, can we electronically transmit your data from the RBDR registry into the SYNC registry. They're both in REDCap, that backbone that I told you all about, so it's super easy, and it'll allow us, as one of the early clinics in the SYNC registry, to be able to fill up their data very quickly. Everyone else will be entering it by manually by hand, and so it'll take donkey's ages, but Edmonton will fill it up with 1,000 patients within the first little while. Yes, another question.
Just to say, we're just going to go into a break, but if you don't mind staying around during break in case there's some more questions. And
well, why don't people grab their muffins and things while I'm talking? If you want to have a question, I'll keep going here if that's okay.
Yeah. Perfect.
Okay.
I was just curious, you mentioned that there was 1,000 patients with their rare bleeding disorders clinic and 500 that are doing SCIG at home.
Yes.
So are the people that are accessing that all patients that are going through K, but are there other people in the city of Edmonton, for example, seeing an immunologist elsewhere and going through separate infusion clinics? Or is everyone being filtered through the K?
That's a simple question with a difficult answer. So there have been issues with capacity that we haven't had the capacity to do them all through the K in a timely fashion. So some of our colleagues have, tried to work around us, but for the most part, probably 99% of the distribution in northern Alberta, parts of Saskatchewan and parts of BC is done through our clinic using this system I've told you about.
And as we continue to find the 70% of people that we don't know yet that have this, like what are the issues with capacity? Like I know there's all kinds of issues with wait times and all of that, but like does your clinic have the capacity to just keep on adding? Is that
keep on trucking?
Keep on trucking,
so
yeah.
The question was, what about our clinic capacity? So let me tell you about our clinic. I followed the model of don't ask for permission, because I spent a decade asking for permission to do things, and nobody ever answered my phone calls, letters, emails, nothing. I got the feeling that was probably purposeful that I was nothing. And so I said, well, we'll just do it. And so we, started with one immunodeficiency patient and one HAE patient. And then a couple more showed up and a couple more showed up. I had no idea how big it was going to grow. So we grew organically. And when I say 1,000 patients, that's 1,000 immunodeficiency patients. We also track 1,000 bleeding disorder patients. And we track probably 500 patients with other problems like hemoglobinopathies and things like that. In the clinic now, we have 4 almost full-time nurses. There's like 3.8 or something like that, and an extra 2 who help fill in. So that's way more than any other clinic in the country. No other clinic in the country has that sort of nursing capacity. In addition, we have 3 clerks. And I've been able to do this because they're covering all the programs, not just immunodeficiency, but HAE and bleeding. And it's been a great model, and they're able to cross-cover each other and stuff like that. But the immunodeficiency population is growing very fast now because there's new technology. There's a thing called bifunctional monoclonal antibodies for treating certain cancers that are fabulous, but they make everybody have low antibody levels. And so as that treatment is taking off, it's creating more patients that we need to treat. So it's an ongoing battle. It'll never be solved. Certainly if I retire in 2 or 3 or 4 years, the problem will still be there, but we'll be way, way, way ahead of the rest of the country in solving that problem and really putting control of your illness in your own hands, which I think is the model that we're trying to follow. I don't know, you guys need to get your coffee.
We've got one question.
Okay, one question off the internet.
Yeah. Okay. Is there succession planning in place if you're looking to slow down in the future? There does not appear to be a lot of options available for patients.
Well, yes. The question is succession planning. What happens when I get hit by a bus? And it is a big problem. Interestingly, when I came to work in Edmonton, soon after they developed a program to let people know how well they were doing academically. As an academician, an academic doctor, I'm supposed to be writing papers and things like that. And I've never been really very good at that. And they've always been on the cusp of firing me. In fact, in the early days, they did try and fire me for a couple of years, but I stuck around, so they put up with me. Eventually, I got promoted to an assistant professor and a full professor. So who knew, right? But I was doing all these programs myself 8 years ago, and it was just overwhelming. So I handed off the hemoglobinopathy program to other doctors who've now added a third in to do, what was one small part of what I was doing. The hereditary angioedema program is completely taken over by Dr. Adatia. The immunodeficiency program, now we have 4 doctors and we're adding a 5th in January. So the chance of you actually seeing me now, if you're a new patient, you won't see me because I'm not seeing any new patients. They're all going to the other doctors as I'm trying to gradually turf my patients to them. So those of you who see me will, one day someone will call you up and say, and you'll say, well, where's Dr. Ritchie? And they'll say, we don't know, he didn't show up today. But yeah, there are a lot of doctors involved in the clinics, and there are people to look after all the individual programs. The bigger question is, will there be somebody with all the interests that I have that will look after the whole rare blood disorder clinic? And right now, there's nobody that's interested in blood product tracking like I am. So I'm trying to convince some of the young guys how critical it is, and that's sort of working. But a more important part of it is to get it to write that into the DNA of Alberta Health Services. In other words, have the nurses and the nursing administrators say, well, this is our standard of care. So have the nurses say, you either have to track your products or you can't be on the program. Because doctors are doctors and we're all a little bit different, have different interests and stuff. So anyway, long answer to a short question. Any more?
That's it. I just want to say a big thank you to Dr. Bruce Ritchie for joining us. Thank you so much.
Thank you.